Hello guys, hope you're enjoying your snow day, or multiple snow days at this point. Today, we're going to learn about Gregor Mendel and Gregor Mendel's studies using genetics. And then in the second part of the lesson, which is actually a separate video, which we'll have to play the second link, we're going to learn how to do simple Punnett squares. I would actually recommend going through the PowerPoint first, and then looking at the guided questions, which are on the packet that has the mitosis animation on it. It's just easier. After the packet's filled out, you can just go through and answer the questions. That's what I recommend. But, alright, here we go. So Gregor Mendel was a German monk. He lived in a monastery, and I guess he was really bored. So he studied traits in peas. So what Mendel did is that he mated pea plants, and we'll talk about how they do that in a second, and sort of looked at certain traits that pea plants had, and sort of saw how they changed through generations. In the next couple slides, we'll take a look at some of the traits that he looked at, but he essentially mated pea plants and then planted the seeds, and then sort of see how the traits changed over time. He collected lots and lots of data, and using that data, made some conclusions about inheritance. So pea plants were a good choice for Mendel to use for his research. First of all, the traits that they have are very easy to see. So you have traits such as flower color, and seed shape, which are obviously very easy to record and recognize. The second reason why pea plants were a good plant to use for research is that they're easy to control their mating. So every plant has both male and female structures. On a flower, the inside of the flower is usually the female part, and the outside of the flower usually has things sticking up which are called stamens, and they produce the pollen. So since pea plants have both male and female parts, they can self-fertilize. Mendel could also control which plants mated with who, because he could take a stick and stick it into the pollen, or grab pollen from the plant and get pollen on a stick, and then bring it over to the female part of another plant and cross-pollinate the two plants. So he could very easily control who was mating with who. Wasn't if you have animals, for example, you can't always control who mates with who in that case. So like we said before, plants were good because they're simple. They're very simple genetically. So most of their traits that they have are controlled by only one gene. And then there's only two versions of the gene. So here, if we see with flower color, there are two, and this is going to come back in this PowerPoint again and again. There are two versions. So there's one gene, and the gene controls flower color, but there are only two versions of the gene. You can either be white, or you can be purple. And because having only two versions of a certain gene makes it just easier to track, as opposed if you have many versions, such as height or things like that, short, tall, medium, in between. But having tracking traits that only had two possibilities sort of made his studies easier. So these are just some of the traits that he tracked. So color of flowers, color of seeds, shape of seeds, color of pods, and so forth. But the thing that all these traits had in common is that there were only two forms of the trait. And that made it very easy for Mendel to track sort of the traits that appeared in the next generation of peas. So here, we're going to take a look at an example of what Mendel did. I'm also going to learn some vocabulary while we're going along. So first of all, Mendel bred pea plants. So here, if you take a look, he's taking the pollen from a white flowered plant and allowing it to pollinate a purple flowered plant, and then the seeds develop in the pod, and he planted these seeds to get flowers. So, and these vocabulary words are going to come back again. So the first sort of mating is called the parental generation. And then the offspring that they produce is called the F1 generation. And if you allow the F1 generation to self-pollinate, or pollinate themselves, then you get the F2 generation. So the parents are the first mating, then the offspring of the parents are called the F1 generation, right here. And then if you allow the F1 generation to pollinate themselves, so this flower will pollinate that one, then you get the F2 generation. So we're going to take a little closer look at the results of the cross that we looked at the previous slide. So in the cross, he took true breeding, purple flowered peas, And what makes something true breeding is that if you mate it with another purple flower, it will only produce purple flowers. And then he took a true breeding, similar thing, this in case it's white flower. And if you mate the white flower with only white flowered plants, then it will only produce white flowers. That's what makes them true breeding. So he mated them, took the pollen from one and transferred it to the other, and he noticed that all the white colored flowers disappeared. And he got, after the mating of these two, the purple with the white, he got 100% purple flowered peas. And Mendel was kind of confused by this. Okay, and this was the F1 generation. So the parents were true breeding purple and true breeding white, and they mated, and they got the F1 generation, which was all purple. Then he allowed the F1 generation to self-pollinate. Okay, and he noticed that the white flowers came back, and they got about 75% purple flowers and 25% white flowered peas. And then Mendel sort of asked this question, like, so what's going on here? So why does the white color sort of skip a generation? And then, so we're going to talk about sort of the math behind this and sort of what Mendel discovered after doing many meetings of this type to determine, like, why is this the case? So why does the white flowered trait skip a generation? So one of the things that Mendel concluded from his research is that traits can come in alternate versions. And he called these alternate versions alleles. So for example, in the previous discussion that we were talking about, the gene was for flower color, but there were two alleles, purple and white. And just to bring, now Mendel knew nothing about DNA or anything like that. But just to bring it back to the DNA, what makes these two alleles are different sequences in the chromosomes. So Mendel knew nothing about that. Mendel knew nothing about DNA. But just to relate it back to what we've been learning for the past, 2 months in molecular genetics, is that what makes these different alleles are the A, T, G's, and C's at a location on the chromosome. And these chromosomes here are actually homologous because they share the same genes. Homologous, remember we learned yesterday or whatever day that was when you're watching this, means that there are two genes, you get one from mom and one from dad, and they share the same traits. So I'm sorry, the same genes, but it could be a different allele. So here, this is the trait, this is the gene for flower color, but this chromosome has the purple allele, and its A, T, G's, and C's are different than the A, T, G's, and C's for the white colored allele. And in actuality, it's more than just the A, T, G's, and C's. The A, T, G's, and C's, when they're different, produce a different protein, and that produces a different color. So just to summarize here, alleles are two different versions of the same gene. In the case that we've been looking at, the gene was flower color, but the two alleles were purple and white. On this chromosome here, just to sort of recap, and I noticed this didn't pop up, these are homologous chromosomes, and we have different versions of the same gene on the chromosome. The purple allele and the white allele. In all diploid organisms, and diploid organisms or organisms that have two copies of each gene, there are actually some organisms that live their life in the haploid state, but we're not going to talk about them. So diploid organisms inherit two sets of chromosomes, one from each parent. During fertilization, you'll get one copy of one chromosome from your father, from the sperm, and one chromosome from your mother in the egg. And when those cells fertilize, you'll have two copies of each gene, and perhaps you'll have two different alleles or one allele. But of every gene that you have two alleles, one from each parent. They may be the same allele or a different allele. So, like for example, for the eye color gene, you may get the brown-eyed allele from your mother and the blue-eyed allele from your father. And now sort of the question is since you have both the blue-eyed allele and the brown-eyed allele, which allele is going to make your eye color? And that's sort of what we're going to talk about next. But Ralph here is asking a question, and the question he's asking is sort of what are the advantages of being a diploid organism? And the advantage of being a diploid organism is that you have two copies of each gene. So if you have two copies of each gene, that means you have a backup copy. So if something goes wrong with maybe your copy from mom, you have a backup copy from dad. Organisms that live their life in the haploid state don't have their luxury. If they only have one copy of each gene, because they're haploid, then they don't have a backup copy. So that's sort of the advantage of being diploid. You have have two copies of each gene, so if something is wrong with mom's hemoglobin gene, and you have a correct hemoglobin gene from dad, then that can make up for the incorrect hemoglobin gene you got from your mother. So back to the white-purple flower situation. What Mendel concluded is that some traits or some alleles mask others. So in the purple-white situation, one possibility is that purple and white blend and produce a light purple. But that's not what happened. The purple in the first generation, after you mated the parents, the white completely went away. So in that case, the purple masked the white gene. And he called the genes that mask, or the alleles that mask other alleles, the dominant allele. Okay, and the traits that are masked by the dominant allele is called the recessive allele. So eye color is sort of an example of that. Brown eyes is the dominant trait, and then blue eyes is a recessive trait. So if you have the brown-eyed allele and the blue-eyed allele, you got this. Let's say you got the brown-eyed allele from dad and the blue-eyed allele from mom. The brown-eyed allele is going to be dominant over the blue-eyed allele, and you'll have brown eyes, or at least dark color eyes, not blue, unfortunately. So once again, over here we have homologous chromosomes, one from mom and one from dad that have the same genes. Let's say you got the plant got the purple allele from the dad plant, and the white allele from the mother plant. Because purple is dominant over white, the plant will have purple flowers, not white. So these are words I've kind of been using in class, and I think some of you have gotten them and some of you haven't. But we're going to finally officially define them. So the first word is phenotype. Okay, and phenotype is what the organism looks like, what you can actually see. Okay, so purple flowers is the phenotype of the flower, what it looks like. The genotype, which we've sort of been using, and I don't think many of you haven't got that, is the genetics, so the alleles that it has. Does it have two purple alleles? Does it have a purple and a white allele? So the genotype is its genetic makeup, and the phenotype is what it looks like, what you see. You can't actually see the genotype. So the phenotype is what you see. The genotypes are the alleles that it has. So if we look at this, picture over here, and just talk about it specifically in terms of phenotype, and then we'll get back to genotype. We have the purple phenotype plant mating with a white phenotype plant, and the purple is the dominant phenotype, and white is the recessive phenotype. And then when you mate them, you all get the dominant phenotype of purple. We're now going to go take a look at what the genotypes of these flowers are. So in Mendelian genetics problems, we represent alleles with letters. The dominant allele is given the uppercase P, and the recessive allele is given the lowercase p, or whatever letter you choose. So flowers that are true breeding, okay, purple flowers, so they're true breeding, they only can produce purple flowers if mated with another purple flower, have two uppercase letters. Okay, so they have both purple alleles. Okay, and then the white flowers, true breeding white flowers, have only little p's. Okay, now in the cross that we're looking at the last page, we crossed a true breeding purple flower with only big p's, and this is the genotype. The genotype is the fact that it has two uppercase or two dominant alleles, and we're crossing it with a recessive, completely recessive plant that has only recessive alleles. Now, because of this mating, and you'll see what you get, okay, you get all purple flowers, okay, that have one dominant allele, okay, and one recessive allele. Okay, that's the genotype of this purple flower, is that it has one dominant allele and one recessive allele. That has a name that we're going to talk about in a second. But the reason why these flowers are purple is that the purple allele, okay, is dominant over the white allele. So there are three different genotypes you can have. First, we're going to talk about the word homozygous. Okay, and homozygous means that you have the same alleles. So you can be either homozygous dominant. This guy is homozygous dominant because it has two uppercase p's, and this one is homozygous recessive because it has two lowercase p's. All right. Then we have heterozygous, okay? And heterozygous, okay, means that you have different alleles, okay? You have one uppercase p and one lowercase p. These words are really, really, really important.